Good morning, everyone. Welcome to day 2 of the Brain Cell Atlas from Data to Knowledge workshop. My name is Eunyoung Kim. I am a program officer at the NIMH and also for the Brain Initiative. I also serve as a co-lead of the Brain Initiative team data together with John Monaco, who will be moderating a later session today. For those who may not be familiar with the Brain Initiative structure, the Brain Initiative team data oversees and coordinates the Brain Initiative's data ecosystem, including all of the Brain Initiative data archive, informatics projects, and the brain knowledge base innovation domain, all of which are highly relevant to the overarching theme of this meeting, especially today's topic on data ecosystem and data to knowledge pipeline. So as you all are, I'm very much looking forward to another engaging day of discussion and fresh ideas on how we can better share, harmonize, and use the Brain Cell Atlas to maximize the impact for the broader research community beyond the Brain Initiative. So we have a packed and very exciting agenda planned for you today. As a housekeeping note, this workshop is intended to be a two-way conversation between the panels and our attendees. So please share your questions or comments using the Q&A feature at the top of your meeting screen. But if you join the meeting via an internet browser, you may not see the Q&A function. So if it's unavailable for any reason, please don't panic. Simply raise your virtual hand and then we will call on you. So before we begin today's main session, we will start with a brief recap of day 1. To do that, I am pleased to welcome Dr. Aparna Bhaduri from UCLA and Peter Koo from Cold Spring Harbor Laboratory, who will summarize and highlight the key themes from day 1. Aparna and Peter, please take it away.
Thank you so much. I'm actually here with Rahul Satija, who also was co-moderating the first panel, where we were really talking about taxonomies and ways for unifying taxonomies and applying them with community standards. I'm hoping that Rahul is here and is able to chime in. If not, while he gets started, I will go ahead and begin with the summary. I think that some of the main themes from the first panel session, we're really thinking about how to collaboratively construct a reference, using this for cross-species analysis and linking across development, and then thinking about how to collaboratively create the reference atlas with a community and leverage spatial technologies to really inform the distribution of said atlas.
Yep, I think I'm online now, Aparna. So why don't we can go back to our previously scheduled plan. I'm just going to share the agenda that we put up yesterday, just so you guys can see the speakers that we had present. But yes, Aparna beautifully summarized kind of our goal is how do we build this atlas, and then the second is how do we apply it in lots of different contexts. So I'll summarize our first speaker and then turn it back to Aparna. So our first speaker was Scott Daniel from the Allen, who talked about the Map My Cells tool that he's built. One of the biggest kind of highlights and takeaways from me was just how advanced and mature these tools are compared to what they were just a few years ago. We've been talking about building these atlases for a long time, but we're actually at a place now where not only do these reference atlas exist, but there are tools that can be very easily adopted and applied across labs and consortia to be able to put things into an integrated and common framework. And there's even sort of community-wide acceptance of some of these, and that's just a really exciting advance that I thought was really a milestone for BICAN and your community. Some of the questions that came up in the discussion were really interesting. Some of them we've talked about for a long time, like how do you sort of decide what's a cell type and what's a cell state, and how can we use other types of modalities and other data types to draw this distinction. And one idea which came from a couple of people, including Bing Ren, is the idea of not just defining a cell based on its steady state sort of marker genes or based on sort of a steady state piece of information, but also sort of its broader gene regulatory network and the pieces of information across modalities and kind of how they work together in concert with its function. So those are harder things to collect at scale, but there are increasingly sets of technologies that enable us to collect both epigenomic and functional data, and thinking about how to integrate those in to the definition of cell types and cell states is a fun challenge going forward. And then there was some good discussion about the pluses and minuses of different types of ontologies and hierarchies. Hierarchical frameworks are really useful for being able to divide annotations at different levels of resolution. But sometimes cell type relationships can be complicated and not beautifully fit into a hierarchy. And we're starting to figure out as a community kind of when one type of structure is appropriate and when others might be as well. So that's my short summary. Aparna, I don't know if you want to add on to that and then talk about our other speakers as well.
Great summary. And I think that that really feeds well into some of the more complex use cases of how we can leverage these references. Fenna Creenan, who's an assistant professor at Princeton University and Neuroscience Institute, gave a really fascinating talk about some of the practical challenges of thinking across species and across space, and questions of how do you have the right resolution, completeness, and then how do you decipher whether differences between species are because of technical artifact or biological artifact. And in many cases, you're seeing these many-to-many cluster relationships, and you can do things like port spatial registration to through another species if there are samples that are available from one context to another. And this is particularly when thinking about human and non-human primate, where sample accessibility and sampling is not always going to be as thorough as it might be in other model organisms, such as mouse, where you can really get it every few hours if you wanted to. In a parallel talk and discussion, and we kind of unified those discussions a little bit, Tom Nowakowski, who is an associate professor at the University of California, San Francisco, gave a really nice talk thinking about the lineage relationships and how progenitor cells in development are following these differentiation programs, and some of the challenges and opportunities associated with annotating and deciphering the meaning of these cell types, and how in some cases it's very clear and in some cases it's not as clear, and how there are some experimental methods that are emerging, such as lineage tracing approaches, as well as comparative studies that go back to the species-species comparison that are really able to fill in these gaps. But ultimately, there's still a lack of resolution that's going to allow us to perfectly link all of the developmental cell types to the adult. And so thinking both about the question of species-species comparison and development, one of the unifying themes of the discussion was that we do need more high quality ground truth data. I think that that's a really good argument to continue the efforts that have been so productive so far. And also thinking about how do you know what are the right marker genes, and how you can really think about which ones are meaningful in terms of deciphering cell types versus which ones are biologically meaningful, and how those change over developmental time. But that there needs to be some method development and also integration of things like gene regulatory networks, as well as some acknowledgements that the different use cases may require different ontologies, and we might need some more complex frameworks in order to account for all of the variation and all of the use cases that are being pursued by the community. I'll turn it back to you, Rahul.
Great, thank you. That was wonderful. And then our last speaker, who I'll summarize briefly, was Xiaowei Zhuang, who addressed spatial technologies and data integration. Xiaowei presented a beautiful new technology her lab has developed, which really aims to be able to identify differences either in short genes or to quantify short genes where you can't have a lot of probes to boost signal in fish-based assays. Or even to be able to extend those profiling technologies to look at one isoform versus another, using a method of in situ RNA amplification that I thought was very exciting. So the discussion focused kind of on two really interesting topics. One is sort of what's the next frontier in spatial analysis. We can do transcriptome-wide imaging now, we can do it at scale, kind of what's next. And Xiaowei talked a lot about her efforts to combine these types of molecular measurements with functional assays, including CRISPR perturbations. If we can start to understand the function of cell types in concert with their spatial localization, we can do a better job of understanding when are small molecular differences between cell types that will always exist due to spatial localization. When are they meaningful? When are they functionally important? Which was a key question. And then a second area of discussion talked about the importance of common coordinate frameworks for being able to register multiple spatial data sets together in an anatomical context as opposed to just a molecular context. Obviously, the Alan has done extraordinary and pioneering work in the mouse, but this is a little bit harder in the human, especially given the phenotypic variation present in human samples. So one of the things that it's not a solved problem yet, but that may make that problem a bit easier going forward, is that actually molecular data is extremely useful for being able to identify, even in the absence of detailed imaging, where different brain structures are actually located, not define them based on sort of histology, but based on actually the spatial co-localization of multiple molecularly defined cell types. So we have more information now when we're trying to build a human common coordinate framework than we had when trying to build a mouse framework. And that may facilitate solving what is actually a much more difficult problem. Yeah, so that's hopefully a good sense of what were some of the topics we covered yesterday. And my main takeaways were, again, just how advanced and mature some of these approaches are compared to where they were a few years ago, and this broader need for sort of complementary and ground truth data to really even further move the field forward.
Yesterday, we had a second panel, panel 1-2, on challenges and opportunities in leveraging AI and ML advancements. I was a moderator with Honghui Zhang, who is currently unavailable because she's traveling today, but she'll be joining later. And I want to first off thank the note takers. I'm going to butcher your name, so I apologize in advance. Mike, Haolizik, Hanking Liu, and Go Cheng Yuan for your fabulous notes. It really helped me to distill what I'm going to cover right now. Overall, the goal of the panel was to examine how AI is currently used across sequence models, single-cell models, multimodal integration, and interpretability in the context of brain cell atlases, and to identify the directions most likely to drive biological discovery next. And the discussion reflected a field that has reached a point of operational maturity for many atlas-scale tasks, including integration, annotation, and prediction. Attention is now shifting towards questions that require richer biological grounding, such as causal inference, functional interpretation, and integration across modalities, spatial context, and biological scales. The panel focused on 4 closely connected areas: how data are generated and labeled, how model performance should be evaluated, how multimodal and spatial data can be integrated in the presence of missingness and imbalance, and how predictive models can be linked to causal and mechanistic understanding. Overall, the panel underscored how choices in data generation, evaluation, and modeling interact to determine the scope of biological insight that AI methods can deliver. And I'll walk through the main sections and highlight the key ideas and future directions that came out of this discussion. So the first section was on where are we now with our AI modeling capabilities and readiness. And we posed a question to our panelists. Where are we now with AI models for brain cell atlas tasks like classification, integration, and prediction? And the two panelists were David Kelly and Fabian Thies. The first section of the panel was meant to ground the conversation, to take stock of where AI models actually stand today for brain cell atlas tasks, and to separate what is already working from what still feels out of reach. And from the sequence modeling side, David Kelly emphasized that these models are already very strong at local prediction problems. They can accurately predict epigenetic marks, chromatin accessibility, gene expression from sequence, and in many cases, they can resolve subtle differences across regions and closely related cell types. However, he also stressed that this success has limits. When biological differences are subtle, there may be very little signal for any model to learn from. In those settings, improving data quality matters more than increasing model size. He also emphasized that identifying causal genes at the relevant cell state, especially at high resolution, remains open challenge. Now, from the cell modeling side, Fabian Thies emphasized that many classic single-cell tasks are no longer the bottleneck. Cell annotation, batch correction, and dataset integration are largely solved at scale and already embedded in standard analysis pipelines. What has changed recently is not algorithmic novelty, but how models are used. And the field is moving towards Atlas scale foundation models that act as reference resources, allowing new datasets to be mapped, transferred, and compared across modalities, systems, and even species. A key point Fabian made is that cell number alone is no longer the limiting factor. What matters is whether a model has seen the right conditions, perturbations, and diversity of states. He also raised the issue that, unlike large language models, we don't yet understand scaling laws in single-cell biology. In many cases, simpler models perform as well as larger ones, highlighting the need for power analysis style thinking to guide experimental design. And across both perspectives, a shared theme emerged. Since biological data is usually collected to answer specific biological questions, not to optimally train AI models, this creates a mismatch between experimental design and modeling needs. So implication going forward is what type of data we need is still an open question, but genetic diversity, perturbation data, and dynamics are all important sources to sample from moving forward. And AI experiment loops can help navigate this space, guiding experiments in an active learning approach. In the second section, we covered molecular and epigenetic data for functional inference. And the question was, how can molecular and epigenetic data be used to infer gene programs and predict functional impact of genetic variation? The lead panelists were Jian Zhou, Mark Gerstein, and Ji Liu. And so from the sequence to function perspective, Jian Zhou discussed how current models are effective end-to-end predictors of variant effects, while emphasizing that variant interpretation is fundamentally a causal problem. He noted that these models operate as black box predictors. They do not explicitly represent the regulatory processes in between, and predictive accuracy should not be conflated with a mechanistic understanding. Bridging this gap will require additional data and validation. Mark Gerstein emphasized the importance of bridging population genetics with AI modeling. Genetic variation across individuals acts as a natural source of perturbation and provides signal that is difficult to obtain otherwise. Linking AI models to classical statistical genetic frameworks such as QTLs, GWAS, and case control studies adds rigor and improves sensitivity and power. Mark also highlighted the central role of metadata. Labels, donor information, conditions, and experimental context often determine whether a data set can be meaningfully used by models. And this is especially critical in the world of agentic AI, when this information when these models are going to be automating data analysis, providing much needed context. And Ji Liu pointed out several gaps that limit current modeling efforts. Protein and RNA modifications remain underrepresented despite their likely importance. Even when epigenetic and transcriptomic data are measured in the same cell, joint modeling can introduce bias if biological dynamics are not aligned. Spatial information is highly complementary to molecular data, but is often missing or treated as secondary. And an implication going forward is, for this section is inferring gene programs and variant effects is fundamentally a causal problem. Progress will depend on integrating population scale variation, perturbations, temporal structure, spatial context, and high-quality metadata into model development. Treating these data types as core infrastructure will allow AI models to move from prediction toward biological insight. And the third section was on benchmarks and evaluations, CASP-like challenges. And the question we posed for our panelists were, how should we define benchmarks and are CASP-like challenges appropriate? And our lead panelists were Ziga Absek and Rahul Satija. This section focused on how progress in AI for brain cell atlases should be evaluated and whether community-wide challenges similar to CASP are appropriate at this stage. Now, from a benchmarking perspective, Ziga emphasized that evaluation matters more than introducing, let's say, the next model. Without standard benchmarks, the field lacks a common notion of progress. At the same time, he stressed that brain biology is much harder to evaluate than domains like protein structure. There is no single correct output, no universal metric, and available benchmark datasets are often limited, biased, or noisy. High sample-to-sample variability often complicates direct model comparisons. Because of this, Ziga argued against relying on single end-to-end like a metric or a score. Instead, he advocated breaking complex biological problems into intermediate tasks with biologically meaningful metrics. Even when the problem is difficult to evaluate cleanly, progress can still be made on well-defined subproblems, such as components of regulatory variant interpretation, provided those tasks reflect real biological questions. From a single-cell perspective, Rahul and Satija emphasized that different areas in AI and biology sit at very different stages of benchmark maturity. Protein structure prediction represents a mature case with clear ground truth and metrics that align well with biological quality. Cell annotation and integration are more intermediate. They have improved substantially, but ground truth remains uncertain or context dependent. Perturbation effect prediction is still early with noisy data, unstable metrics, and little consensus on evaluation standards. Rahul cautioned against moving too quickly toward cast-file competitions, and historically, methods are adopted because the community trusts them, not simply because they win benchmarks. So trust is built through robustness, interpretability, and repeated success on real biological problems. Benchmarks can support that process, but they do not replace it. Across both perspectives, there was strong agreement that benchmarks must be tied to biological goals. Metrics that are easy to compute are not necessarily the ones that matter. The field won't benefit from shared benchmark data sets and standardized, or the field would benefit from that and standardized processing pipelines, if only to avoid fragmented comparisons where every study defines its own evaluation criteria. Now, an implication going forward is progress in evaluation will come from alignment rather than competition. The priority is to define shared biologically aligned tasks and intermediate metrics that the community can trust. CASP-like challenges may become useful later, but only once evaluation criteria reflect biological relevance and are grounded in real use cases. Now in section 4, we covered relating genes and cell types to brain function through multimodal integration. And the question was, how do we link molecular profiles, anatomy, and function across space and time? The main panelists were Riza Abbasi, Trey Decker, and Viren Jain. Reza emphasized that off-the-shelf foundation models are useful starting points, but rarely sufficient for specific biological questions. Real applications typically require fine-tuning or task-specific modeling choices, especially when the goal is interpretation rather than prediction. He also stressed that harmonizing data across labs, protocols, and technologies remains a major challenge, and that pre-trained models alone do not resolve these inconsistencies. This highlights the importance of high-quality metadata that Mark Gerstein mentioned previously. Trey Decker emphasized that multimodal integration is constrained by uneven data coverage across modalities. Different data sets measure different features, often at very different scales, and approaches that rely on the intersection of modalities quickly discard much of the available data. He argued that models need to operate closer to the union of modalities and remain effective when cells or samples have different subsets of measurements. As more modalities are integrated, interpretation also becomes harder. And since multiple embedding spaces can obscure which biological features are driving results. The discussion then turned to spatial organization and the common coordinate framework with input from Viren Jain and others. While a shared spatial reference is clearly valuable for aligning data, several panelists emphasize that its real potential lies beyond alignment. A common coordinate framework can support consistent comparison across data sets, expose uncertainty, and highlight regions or cell states where information is missing or ambiguous. There was also broader discussion about how multimodal integration reshapes our view of brain organization. Traditional atlases impose hard regional boundaries, but higher resolution molecular, spatial, and functional data increasingly suggest graded and probabilistic structure. Multimodal models will need to reflect this continuity rather than enforce discrete partitions. So an implication forward from this section is that progress in multimodal integration will depend less on more expressive models and more on how missingness, imbalance, harmonization, and interpretability are handled by design. Models should be built to work with heterogeneous, incomplete data and to make uncertainty explicit. Spatial frameworks like the Common Coordinate Framework can play an active role in guiding analysis and experiment design, especially when integration becomes bidirectional. Data informs models, and models help prioritize what data to collect next. Now, in the final section, it's on mechanistic interpretability and biological insight. We pose a question: how do we move beyond black box prediction to biological insight? And the main panelists were Jingyi, Jessica Lee, Sudipta Sengupta, and Steinertz. And this section focused on how interpretability fits into brain cell atlas modeling and what kinds of insights are realistically achievable at different biological levels. And Jessica emphasized that interpretability is often used very loosely. Different methods claim interpretability while answering different questions. She highlighted that stability is not the same as interpretability. A feature that appears consistently across resampling does not automatically correspond to meaningful biological explanation. Interpretability only becomes well-defined when it is tied to a specific task in biological context, such as enhancer activity, variant effects, or regulatory interactions. Without that grounding, interpretability claims are difficult to assess or compare. Sudipta discussed recent progress in understanding transformer models by inspecting their internal representations. In LLMs, large language models, examining internal activations has revealed structured intermediate representations that go beyond next token prediction. He described how similar approaches applied to biological transformers have uncovered internal features aligned with known biological structure, such as coding regions, RNA, protein secondary structures, and so forth. The broader point was that even, self-supervised models can learn rich internal representations, and probing the model internals may offer a path towards a deeper biological insight. Steinertz framed interpretability across different levels of organization. At the enhancer level, interpretation is most tractable. Models can reveal motif grammar and regulatory rules, and those rules can be tested through synthetic sequence design and perturbation experiments. At the gene level, interpretation becomes more challenging because of how enhancers combine to regulate expression is not well understood. At the gene regulatory network level, interpretation is hardest and requires carefully designed perturbation experiments and close coordination between modeling and experimentation. Across levels, black box prediction alone is insufficient for building mechanistic understanding. Now, an implication moving forward is progress in interpretability will come from focusing on concrete tasks and specific biological levels, rather than aiming for universal explanations. Jessica drew a clear distinction between causal inference and mechanistic understanding. Causal inference focuses on estimating population level effects under explicit assumptions and represents a realistic early or midterm goal that we can probably achieve soon. Mechanistic understanding goes further, aiming to explain how the system operates in physical or biochemical terms. Even for causal discovery, the tools remain a bit immature right now, leaving substantial open space for methodological development. And so in summary, I'd just like to thank all of the panelists for a wonderful and insightful panel, and the co-moderators, Hanki Lee, for helping out, and the note takers for helping to synthesize what was discussed.